potential for medicine in some of the movies that we'll see. I'm going to show you a couple of short videos here. It sort of gets at this idea. Okay. Okay, so 3 more cells here. One of the diseases of interest is Parkinson's disease. And this involves a certain cell type that's found in an area of the brain known as the substantia nigra. And there are cells in this region that make a critical neurotransmitter known as dopamine. And in Parkinson's disease, it is these dopamine-producing cells that mysteriously die. So you have the rest of the body and the rest of the brain is more or less okay, but these particular neurons are dying. And as a result, some of the classic phenotypes that you see are the kind of tremors and shaking and shuffling gait, small steps, issues with balance. It all tracks back to the death of these particular neurons. And so people have been already exploring in mice and in humans, could we add in cells that are in a more embryonic state and see them differentiate into dopamine-producing cells? And so here's a study where some fetal neurons, so some very immature cells in a status of early differentiation as neurons were added back to the adult brain. And then the red here is detecting dopamine production by these dopamine-producing neurons. And so before the study was done, there was some dopamine production, but after 12 months of adding these fetal stem cells, or new source of dopamine cells, now the dopamine production is increased quite a bit. So, you know, it's kind of early days, but it's pretty exciting to think that this kind of thing might even be possible. Okay, what about limitations of iPS cells in particular, or really any stem cells in general? One is, will they form tumors? So, you know, these cells that are at these really early stages that are pluripotent, I mean, they have an ability to form a lot of things, and that's good, but you know, what if they form the wrong thing? And what if that growth is uncontrolled? That could give rise to a tumor. And there's a type of tumor called teratoma. And in this kind of tumor, you often see differentiated cells of several different cell fates. So you might see some like tooth cells and mesoderm cells and just a bunch of different things. And that would require a cell that was fairly pluripotent that just got out of control in terms of its growth and its properties of differentiation. So there's a risk if you're going around introducing these stem cells that are highly pluripotent in environments that we don't fully understand. Maybe they're going to make tumors. And so I think that we're still kind of at the early stages yet with understanding what exactly is the risk here and how could we control it. Another possibility is variability. How much variation is there going to be from one person to the next? People are different ages, or different genetic backgrounds, different status of health, different status of degree disease progression. To what extent will that work for some and not for others? And then a big challenge is the delivery. So how are you actually going to get these cells to the right place at the right time, or in a status that they could then make connections? Because during development, all this is happening together. You know, all of the different daughter cells are simultaneously building the organ. But now, in an adult, all of that's done, and you're just adding in some newbies and expecting them to try to integrate and be functional, and maybe that's a difficult process. Okay, so now I'm going to pass out this in-class exercise, and we're just going to watch 3 short videos on YouTube about stem cells. And there's a couple of questions here on each of them, take notes of videos. And this is exercise 6, and we haven't done exercise 5 yet, but don't worry, we're going to get to it. I'm not quite sure if I might have printed it out first on this.
But after only a few rounds of cell division, the cells begin to lose this totipotency and to differentiate, sorting into groups that will either contribute to the placenta or to the embryo itself. In this 8-cell embryo, called a morula for its berry-like appearance, all cells express a pair of genes, CDX2 and OCT3/4. But as the cells of the morula go through their next round of division, CDX2 is downregulated in the innermost cells, while OCT3/4 remains expressed throughout. The segregation of these genes is maintained as the cells continue to divide, until after about 10 rounds of division, the embryo has become a hollow ball, known as a blastocyst. The outer layer of the blastocyst is made up entirely of cells that express CDX2. This trophectodermal layer will go on to become placenta. On the interior, OCT3/4 marks a small cluster of cells with phenomenal ability to differentiate. This inner cell mass is made up of cells with the developmental potential to give rise to all of the body's many cell lineages, an ability known as pluripotency. And indeed, every one of the cells in your own body can trace its ancestry back to these few dozen pluripotent stem cells. The vast majority of the body's trillions of cells are differentiated, meaning they have taken on specialized properties and functions, and have lost the ability to generate cells of other types. Skin, muscle, blood, bone, and the nervous system are all made up of populations of differentiated cells. There is another type of cell, however, that remains less differentiated and retains its ability to give rise to other cell types. These cells are known as stem cells. Stem cells exhibit two characteristics not found in more differentiated cells. By definition, stem cells are able to divide and make copies of themselves indefinitely, a capacity known as self-renewal. This endless power of division is not limited to creating more stem cells, however. They can also give rise to a wide range of mature cell types. These unique characteristics make stem cells indispensable, both for replenishing the body's cells as they age and are lost, and for constructing the body itself from a tiny number of cells during development. Even after the embryo has grown into an adult, many of the body's cells continually die off and need to be replaced. For example, billions of red blood cells need to be replaced each day. This is the work of stem cells known as hematopoietic stem cells, which are located in the bone marrow. Residing in a complex cellular environment, the hematopoietic stem cell is able to generate precursors to every type of blood cell. And indeed, these stem cells have been used by physicians for decades to replenish patients' blood supplies following radiation treatment for cancer. The stepwise differentiation of a stem cell's progeny depends on combinations of genetic and environmental factors that gradually steer these cells into following a specific lineage. This process involves different sets of stimulating factors operating in the cell's environment, at its surface membrane, and within its nucleus. Each step down the path draws it closer to becoming a functional red blood cell and entering the bloodstream. The lining of the small intestine turns over even faster than the blood. At the microscopic level, this epithelial surface consists of billions of projections called villi, with stem cell-containing areas known as crypts near their bases. The intestinal crypt contains slowly dividing stem cells that maintain their own numbers while also producing rapidly dividing progenitors. These transit-amplifying cells churn out all of the differentiated cell types needed to migrate upwards and replace older cells as they are shed from the tips of the villi. This constant self-renewal by stem cells is a feature of many of the body's systems, including skin, hair, and bone. Not all of the body's organs regenerate as readily as the blood and the lining of the gut, and the multipotent stem cells that have been identified in the adult body are both limited in their ability to generate other lineages and difficult to maintain in culture. In contrast, the pluripotent cells of the inner cell mass can be harvested and grown indefinitely in vitro, and can give rise to every type of cell in the body. These pluripotent cells in culture are known as embryonic stem, or ES cells. By culturing these cells under controlled conditions, it is possible to steer their differentiation into specific cell types. For example, ES cells cultured on a bed of stromal feeder cells and treated with inductive factors such as BMP4 and sonic hedgehog tend to differentiate into neural lineages. By testing various ES cell culture conditions, scientists are gaining new insights into the biology of the differentiation processes that build and sustain and may one day be used to heal our bodies. Stem cell researchers are currently exploring ways to differentiate ES cells into lineages that are of great medical and biological interest. The nervous system, the pancreas, and the heart all regenerate too poorly to restore themselves after serious injury or degenerative disease. Scientists are now seeking ways of using the knowledge they gain every day from the study of embryonic and somatic stem cells to help save patients' lives and to improve our understanding of their seemingly limitless potential.
Okay. I think I'm just going to head right into the next one since we're almost running out of time.
In 1996, the world's first cloned mammal, Dolly the sheep, was born. Her birth proved that the mammalian genome could travel back in time. Placed into an egg, the DNA of a specialised or differentiated cell gained the powers of an undifferentiated stem cell. Inactive parts of the genome were reawakened and directed the development of a whole new animal. Dolly's birth launched a race not to clone people, but to find a way to make human embryonic stem cells from a specific individual. Embryonic stem cells can make any sort of body tissue, so this promised to provide tailor-made human cells for research and transplantation. But making cloned embryonic stem cells in the lab proved very difficult, and on top of that, in 2005, the field was bruised by a high-profile fraud. Around this time, Shinya Yamanaka finished his own experiments. he found a way to make cells similar to cloned embryonic stem cells, but without using eggs or embryos, making the process much more straightforward. His technique was so revolutionary, he worried other scientists wouldn't believe him. Using just 4 genes, Yamanaka had changed mature mouse cells into pluripotent stem cells, cells that could then be used to make almost any cell in the body. Yamanaka named his reprogrammed cells induced pluripotent stem cells, or iPS cells. At first, the scientific community was sceptical, but once Yamanaka revealed the identity of the 4 genes, other scientists quickly reproduced his work. One of these scientists was Konrad Hochadlinger. He engineered his mouse cells to make green fluorescent protein whenever a crucial stem cell gene was active. Then he tried Yamanaka's reprogramming technique for himself. To his astonishment, his cells glowed green. They had indeed turned into stem cells. Hochadlinger and other scientists showed that these stem cells were capable of turning into all kinds of tissue in mice, including heart tissue, skin, and even sperm. Then came human cells. Just a few months later, more scientists showed that our own cells could also be rewound to a stem cell state. So much for cells in dishes. The ultimate test was to make healthy, fertile mice entirely out of iPS cells. Recently, iPS cells rose to this challenge too. Nature Methods has named iPS cells Method of the Year 2009 because of their promise as tools to study biology. Although iPS cells were first made some years ago, it's only now that they are beginning to be used in this way. Studying the cellular machinery that enables an iPS cell to turn into another cell type could help us understand what makes a nerve cell acquire the ability to transmit impulses, or a heart cell to beat. Researchers are also excited about using iPS cells to study human diseases in human tissue, likely to be a more accurate way to model disease than using mouse models. One of the first studies to use iPS cells to study disease was by Clive Svensson. He made iPS cells from cells taken from a young boy with spinal muscular atrophy, and then turned the iPS cells into neurons. This enabled him to study the differences between diseased and healthy brain cells. There are still a few problems with this approach. Not all cells that look like iPS cells have actually been thoroughly reprogrammed, so researchers are working on better ways to make and assess iPS cells and their offspring. And this work won't spell the end of embryonic stem cell research. Embryonic stem cells are still the best understood pluripotent cells, and it remains to be seen if iPS cells are entirely equivalent to them. But the iPS revolution has most definitely begun. In the last year, labs all over the world have published high-profile papers, and many research groups are starting to make disease-specific iPS cell lines. In the next few years, these new additions to the biologist's toolkit should start to show their full potential, increasing our understanding of human diseases and the basic workings of our cells.
Okay, there's barely enough time to see the last one.
For more than a decade, biologists have been able to turn stem cells into beating heart cells in a petri dish. And bioengineers have had some luck getting stem cells to grow on scaffolds to make simple, hollow organs like tracheas and bladders. But just how hard would it be to make a large, complex organ, like a heart, from scratch? Getting beaten clumps of cells to organize into a three-dimensional heart requires scaffolding. At the Massachusetts General Hospital in Boston, Harold Ott and his team start with the scaffold that nature provides. They're using a technique called decellularization to strip away all the living cells from a heart. The owner of this specimen died only a few days ago. It will sit in a specialized vessel for about a week, while chemicals wash away its DNA, lipids, soluble proteins, sugars, and all other cellular material.
The building, if you want to renovate it or repair it, you go in, you knock down drywall or get rid of cabinets and stuff like that. And basically we're going into the matrix and doing that by perfusing solutions and detergents. We're getting rid of all the cells.
Afterwards, all that's left is a pale, white matrix of collagen and other structural proteins that once held everything together. Stripping away the donor cells removes the antigens that can cause the body's immune system to reject the new organ. The scaffold can then be repopulated with healthy new cells that are immunologically matched to the patient. The team hopes to one day completely reconstitute working hearts from a scaffold like this. But today, they're focused on how they might develop parts of the heart using the same process. They've taken a valve and part of the aorta from a decellularized heart and sewn it onto a cadaveric heart.
So this part here is the aortic root, including the aortic valve and the ascending aorta. And this has been decellularized. So this is a decellularized graft sewn onto a cadaveric human heart. And you can see here the right coronary artery button and the left coronary artery button sewn onto that decellularized graft. And the same thing here, this is the pulmonary artery and the right ventricular outflow tract here of the cadaveric heart. This is the graft, the decellularized graft that has been sewn onto the heart.
Amazing. Harold and his team want to check if their graft would be able to do what these structures do in real life: prevent fluids from flowing back into the heart and feed oxygenated blood from the aorta to the arteries that feed hungry heart muscles. They use a CT scanner at the hospital next door to take a look at their work. Harold pumps contrast fluid through the aorta to check for backflow and to see if the arteries that feed the heart muscle leak. The team knew an emergency department case could come through at any time, so they had to act quickly. Back in the control room, Harold runs through his handiwork with radiologist Brian Goshadra.
What we have here is a CT of this explanted heart, and you very nicely injected the aortic root. We can see that you've sewn in your syringe to this area. Just to orient us, I'm really quickly going to put it into a 3D mode down here, and then window away so we can see what's left. Let's just blow that up. So, when you look at the sizes of this aortic root, does that look physiologic to you? What is the anatomy? Looks pretty close to what I would expect in a physiologic human on this. You can see it's very nice and smooth. So what matters most clinically is the inner luminal smoothness, and it really does look pretty good.
The process of decellularization and stem cell seeding could ultimately develop better replacement parts for the heart. These parts might prove more able to repair themselves and could theoretically grow with a young patient. To Harold, this technique could transform medicine.
Last night we were treating a patient who came in and developed sudden onset heart failure and is now currently on a machine that supports her heart and lungs. So seeing these patients and seeing that clinical need and these patients' families and understanding where all this research funnels into ultimately, which is helping patients and developing tools to improve lives of these patients in end organ failure, that's really what gets us going, I think, and what keeps us going.
Okay. Dr. Towson, this 5 years.
They never said this original source would be Sherman, so.
Yeah.
It was the donor. It was the heart. It was the dead person.
Sorry.
Don't ask questions.
I actually showed you a different video, so don't worry about that.
Okay. I was-